everyone. Thank you for joining us today for the Precision Medicine and Health Disparities, the Promise and Perils of Emerging Technology webinar. Before getting our presentations, I would like to provide a few tips for using Webex so you get the most out of today's webinar. All attendees will be in listen mode only. Please feel free to submit your questions throughout the presentation in the Q&A panel and select all panelists from the drop-down. We will ask them on your behalf during the Q&A portion of the webinar. If you need to view live closed captioning, please refer to the Media Viewer panel. I wanted to note that today's webinar is being recorded and will be posted online at a later date. Please also feel free to tweet today's webinar using #PMPH. With that, I would like to introduce our moderator for today, Debbie.
Thank you, Jennifer. Today's webinar is sponsored by the Precision Medicine and Population Health Interest Group at the National Cancer Institute's Division of Cancer Control and Population Sciences, the National Institutes of Health Genomics and Health Disparities Interest Group, and the Centers for Disease Control and Prevention's Office of Public Health Genomics. Advances in genome sequencing and other omic technologies and big data promises a new era of personalized medicine and public health. However, it's not entirely clear whether the new technologies can be used to understand and address existing population health disparities. This one-hour online webinar explores the intersection among genomics, precision medicine, and health disparities. Our two speakers will explore the appropriate role and impacts of genomics and tools of precision medicine in understanding and addressing health disparities in the U.S. and around the world. After two 20-minute presentations, another 20 minutes will be devoted to a dialogue and questions and answers from both the live audience here and online attendees. I'd like to thank the organizing committee for today's webinar, Dr. Vince Bonham from NHGRI, the Human Genome Research Institute, Michael Hahn, also from NHGRI, Muin Khoury, and myself. Muin is from the Office of Public Health Genomics at CDC. I'd like to turn it over to Michael Hahn.
Great, thank you, Debbie. I'd like to introduce our two speakers today before we begin the first talk. Dr. Charles Rotimi is Chief and Senior Investigator of the Metabolic, Cardiovascular, and Inflammatory Disease Genomics Branch at NHGRI. Dr. Rotimi's research focuses on understanding the etiology of complex diseases and health disparities. His team takes advantage of vast interethnic differences in the distribution of genetic and environmental risk factors, with particular emphasis on populations of African ancestry living in Africa, the U.S., and the Caribbean. Dr. Rotimi is also founder and director of the NIH's Center for Research on Genomics and Global Health, with the central mission of advancing research into the role of culture, lifestyle, genetics, and genomics in disease etiology, differential susceptibility to disease, and variable drug response at the individual and population levels. Our second speaker is Dr. Wiley Burke. She is Professor Emeritus and former Chair of the Department of Bioethics and the Humanities at the University of Washington, as well as Adjunct Professor of Medicine and member of the Fred Hutchinson Cancer Research Center in Seattle. Dr. Burke's work focuses on the ethical and policy implications of genetic information in research, public health, and clinical care. She founded the University of Washington's Center for Genomics and Healthcare Equality that addresses the implications of genomic research for underserved communities and co-directs the Northwest Alaska Pharmacogenomics Research Network, a research partnership involving universities and tribal communities in Alaska, Montana, and Washington. With that, Dr. Rotimi, we hand it over to you.
Thank you for that introduction, and thanks for inviting me to give this webinar along with my colleague, Dr. Wiley Burke. I thought I would start by defining what health disparity is, at least from the way I think about it, and that is the disproportionate number of health conditions and deaths when you compare different groups in the same population or at the global level. And so, push down. Okay, sorry. I was trying to move the slides there. So that's my definition of health disparity that I think sort of captures what we really mean, and that's we are comparing groups and we are trying to understand why we see differences in disease distribution There are many forms of health disparity. You can see health disparity along ethnic or ancestral groups. Sometimes people call that racial group. I hardly use that term. I tend to look at it from ancestry and ethnicity level. So, for example, among the group called African Americans in the United States, which is about 13% of the population of the U.S., but when you look at the rate of new HIV infection, in the U.S., it's about half of the new cases of HIV are African Americans. So that's clearly a health disparity, issue. And you have also have health disparity along age, gender, and again, layer that with ancestry. So the prevalence of hypertension that I displayed there on the right side of the slide, you see that women are slightly healthier when it comes to hypertension. And of course, as you age, again, just about once you hit age 60, just about everybody gets hypertension at that point. And then along ethnic and ancestral group, you see that African Americans, again, have much higher rate, almost double that of the other ancestral group within the United States. So that's clearly a high disparity issue. Another way to look at it from a global perspective is this study that I did with my mentor, Richard Cooper, when I was at Loyola Medical Center, where we looked at over 10,000 individuals from populations that share recent ancestry, what we call the African diaspora. So we have individuals from Maywood, Illinois, from Barbados, St. Lucia, Jamaica, Cameroon, Nigeria. We looked at urban and rural environment. As you will see in that cartoon, you'll see that there's almost a monotonous increase of hypertension rate from rural Nigeria all the way to among African Americans in the western suburb of Chicago, from a low of about 7% to about 35% in Chicago. And among the Yorubas in Nigeria, in the Ibadan area, you also see a huge difference between urban and rural dwellers. So that difference you see between urban and rural dwellers is almost as high as what you see between African Americans and other ethnic groups. So that's clearly not anything to do with genetics. It has to do with the fact that you're living in different environmental settings. And the fourth bullet there, which is socioeconomic, as poor versus rich people. Again, what you see there is a palm wine typer. This was the very, very first scientific paper I published with my BS degree at University of Benin, Nigeria. Then I did not realize that I was actually studying health disparity. This was actually trying to understand why, the implication of aflatoxin, which is a byproduct of this Aspergillus flavus, a fungi that grows on Nigerian beverages, or beverages in general, just because of lost refrigeration, storage. And guess what? Who doesn't have refrigeration is usually poor people. So we're actually doing health disparity research, but then I really didn't realize it, as such, until I came to the United States. This is a very important textbook, a book that just came out by a physician that I've practiced a long time in the Chicago area called The Death Gap. I strongly recommend this book for anybody who's interested in health disparity research, because it really just shows to you how important it is, the fact, the environment that people live in, the way society has structured itself, access to care, how all of that really drives what we see in terms of health disparity. If you look at the Loop, if you've been to the Chicago, you relate to the area I'm describing, which is mostly medium income of about 80,000, so fairly wealthy individuals. And in just a few 5 miles down from there, you look at North Lauderdale, medium income there is about 25. You see the huge difference in terms of, again, life expectancy. And then even more dramatic, if you go to the area of what we call Hyde Park, which is a racially diverse environment, and then just a few blocks down the road to Washington Park, median income drops to about 22, and you see the dramatic difference in life expectancy. So Washington Park may as well be living in Iraq or Baghdad or somewhere in terms of their life expectancy. So it clearly, it shows the importance of the environment and societal structures in terms of health disparity. So I want to end sort of, I really want us to appreciate what are the major causes of health disparity, but I'd like to end that, section of my talk by saying, really quoting what was published not too long ago in the PBS, and that is the wages and benefits we are paid, the neighborhoods we live in, the schools we attend, our tax policies, our health issues every bit as critical as diet, smoking, and exercise. The unequal distribution of social factors that drive health disparities are not natural or inevitable. They are the result of choices that we as a community, as a state, and as a nation have made, and we can make it different. And again, just to quote Charles Darwin, if the mystery of our poor be caused not by the laws of nature, but by our institutions, great is our sin. So clearly, the major drivers of health disparity is really things, the way society has structured itself, the things that we are exposed to within the society. Then you have to ask, why does Charles Rotimi lab study genomics in the context of health disparities? The way we look at it is that it's really, genomics, I think, or genetics is a piece of the puzzle. There's nothing that affects our biology that doesn't go through some physiologic process. And those physiologic processes are under the influence of genetics. So we need to study it, and I'm going to give you examples. Hopefully at the end of my talk, I'll convince you that genomics has a role to play, however small that role is, that we need to appreciate it. So the way I look at genomics, I look at our genome as a history book. This history book has captured our experiences of our ancestors in different environments in a very unbiased way. And we are just learning to read this book. And if we read it and don't put our biases on it, I think we will begin to appreciate our history, our health, wide differential distribution of diseases, at least from a genetic point of view. So, part of my major concern in the work that I do is the fact that the way genomics is currently deployed is that it is not evenly deployed across different ancestral populations. So my worry is that whatever gain that we're going to get from genomics, it may not be equally applicable just because we have not studied different ancestral populations. And as you see in that slide, I say, with tomorrow medicine and technology work for all human beings. So that's a major concern of mine. And because of that, I have participated in major genomic work that brings in different ancestral populations from different parts of the world, like the International Heart Map, where we engage the Yoruba community, the Maasai and the Luo community in Kenya, and also more African populations in terms of the Thousand genome. So all of those are publicly available genomic resources that is ensuring that we apply genomic tools to different ancestral populations, because the way I like to look at genomics is, it's like going to the tailor to make sure your clothes fit. Nobody can represent those genetic variations that exist in the different part of populations. We have to study them, in a sense. So, what is genomics telling us? The first, one of the most important things is really shedding light into our history in a way that we couldn't really do in the past. Our history, anthropology has given us very good information in the past about where we all come from and how we are related and how we migrated to different environments. But genomics is shedding new insight into this relationship. For example, we know that clearly now there's really very little argument that we all started somewhere from Africa before we migrated to different parts of the world. Hence, I use the expression that we are all Africans beneath our skin. And more importantly, when some of us left Africa, we integrate. That's a fancy word for saying we made it with Neanderthal in the Middle East and in Europe and in Asia. And some of that evidence of that different species is actually in our genome now in some of this population, up to about 4%. So the question is that part of the genome important in terms of disease, in terms of health, in terms of other things? So we know it's important in terms of history because it's shedding new insight. So there are some of the things that we're learning in terms of what genomics is helping us to understand. Also, genomics is helping us to show how we are all related. This is a study that we published from my lab where we looked at the ancestry of about 6,000 individuals from 30 major language groups. where we show very clearly that we have over 21 ancestries. And the majority of us, over 97%, have mixed ancestry. And I'm sure you've seen some of these commercials from 23andMe on TV where people are surprised that, oh, I'm also that, oh, I'm also this, because basically we've been looking at ourselves from our skin color or some of the little oral history that we, but genetics is sharing new insight into that in the way that we are, for the very first time in our existence, we are appreciating the fact that we are a mosaic of different things, and we need to appreciate that history. And that is why it's extremely difficult to use genetics to define race. We have one human race. If you try to use genetics to define the racial groups or define different groups, you're going to run into this problem of trying to separate soup. No matter how you cut soup, the soup stays mixed. That is genetics. And that's what genetics is telling us about how to define our racial groups. So we can continue to use that word in a very colloquial sense and all of that. But when it really comes to using genetics to understand that, it becomes absolutely very difficult without creating exceptions and outliers. So the other thing genetics is really telling us is providing insight into health disparity. For example, there was a recent paper in the Indian Journal of Medicine and this editorial in New York Times that says that we are misdiagnosing heart disease in African Americans because the reference panel that we are using is not comprehensive enough. We don't have enough controls from African Americans in that panel, so we are calling things that are not deleterious, deleterious. So you can see how that can indeed increase somebody's anxiety when you're telling them they have something that they really shouldn't be worrying about. It also helps us to understand biology. By looking at different ancestral groups, we are able to see things that we would otherwise not see if we just focus on European ancestry individuals, for example, like this particular drug and gene for cholesterol, LDL, the PCSK9, which is a very interesting variant that helps to lower, when you inhibit that gene, you help to lower LDL. And we're able to find it because we're looking at African Americans, where it's about 2%, and it's very, very low in European populations. So you have more power to see it when you look at African Americans. So the other example there is a study of genetic screening for hyperreactivity to an HIV drug. This was very important when it first came out because originally, if you look at just African Americans, this variant is about 1%. Among Europeans, about 3 to 5%. And there was this thinking that because it's so low in African Americans that we don't need to screen African ancestry populations for this hypersensitivity for HIV. But if you look at the Maasai in Kenya, they have one of the highest rates in the world. And if you go just down the road there and go among the Yorubas, it's almost zero. So using the label Africans completely distorts the picture of this genetic variation. Hence words like African, words like European, and are really not very helpful when it comes to treating individuals, which is usually a sample size of one, you and your doctor. And even sometimes your mom cannot represent you in that instance. So it's very, very important for us to appreciate these differences and what genetics is bringing to us. And this is what, again, when we say precision medicine, this is really, in a sense, what we are talking about. This is another important variant. This gives us an example of geography, evolutionary biology, and ancestry. How those things come together to provide insight into kidney disease in African Americans and African ancestry populations. So it is known in the U.S. that African Americans, about 13% of U.S. populations, which I said earlier, but 32% of kidney failure occurs among African Americans. So why? Physicians have been scratching their head over the years, and it turns out that there's a particular gene and a variant of a gene called APOL1, and you can maybe read about this later on. It turns out that this variant really does put people, if you have 2 copies of this risk variant, it puts people at very high risk for getting kidney disease and going into kidney failure. And the other important thing is that it is common in African Americans and African ancestry populations. more importantly is the evolutionary biology concerning this condition, because it came as a result of, to a high frequency, because it's actually protective against sleeping sickness in some parts of Africa, specifically West Africa and some parts of East Africa. So you can understand that under that environment, sleeping sickness and trypanosomiasis was a major killer, devastating disease. So the genome evolved to protect against that. But when you're now in an environment where that's no longer the problem, the cost of that protection is kidney failure. So again, a very, very important, condition. That's some of the insight that we're gaining. The other thing, again, like I said, is that in terms of health disparity, we are not applying genomics well enough. And again, I just want to make that point. As a result of that, we funded the African Society of Human Genetics, which now helps us to institute what we call Human Hereditary and Health in Africa, H3 Africa, which is revolutionizing genomics across the continent of Africa and is bringing novel insight into genomics in that part of the world. So the cartoon you see on the left is before H3 Africa. African investigators, usually they didn't talk to each other, they were following the money, but H3 Africa has turned that, and people are now interested in genomics and helping us to understand whatever insight genomics can bring is important. A very good example of that is the recent Ebola epidemic. Part of the funding from H3 Africa helped to create this center, African Center of Excellence for Infectious Disease in Nigeria. And because that infrastructure was there, they were able to quickly sequence the Ebola virus and able to make that available. They were able to track how the virus is evolving over time. So clearly, that, again, is very important. That is why we have to make sure that as we are using genomics to understand disease, that we are creating infrastructure in different populations so that we can understand and put the right infrastructure in place. The other point I want to make is, again, with regard to sickle cell, this is the first molecular disease that we know about. And it is actually disheartening that up to now, we still have very, very, what I call less than ideal treatment for sickle cell. But the new technology in terms of what we call CRISPR, which is basically gene editing, is now providing a way for us to see if we can actually go into the genome of these individuals and edit the gene in a way that we can really cure this disease. So again, one way that genomics is going to help us in a very direct way to impact health disparity in this particular instance of sickle cell disease. Sickle cell disease is truly, again, a genetic disease. So I'm going to skip some of these slides, and the point I want to make here, again, is how genomics is helping us to appreciate the fact that we are indeed a mosaic. And this is an example of how, when you use race in the context of medicine to treat people, you need to be very, very careful. This was an 8-year-old boy, phenotypically European, presented with acute abdominal pain and anemia. And surgery was being considered, but it turned out that this young boy had sickle cell, and that was not obvious just by looking at the kid. And again, what turns out is that his parents were from Grenada and were of Indian and Northern European and Mediterranean ancestry. So you can see where the sickle is coming from. But by looking at this individual, you cannot tell. And the cartoon on the right also points out, again, I said earlier that APOL1 gene is indeed very, very important in terms of kidney disease. And I said African and African ancestry individuals. This picture, you see that these are individuals who identify as Hispanic. But some of them carry this variant. Why? Because of African ancestry as a result of the slave trade and the mixing of people. So it's very, very important for us to appreciate this. And I'm going to end with this particular slide here and to say that in this era of precision medicine, we have to be accurate, we want to be accurate, and therefore all of us, we have to participate in genomic research. You cannot depend on my measurements, otherwise the clothes you wear may not fit. So it is very important for us to be part of this effort and to make sure that this happens. And we have to ask the question, who is Black? Is it Tiger Woods? Is it Naomi Osaka, Haitian father and Japanese mother? So we need to be careful how we implement precision medicine to make sure that we are doing it correctly, and we need to guide against promising too much. There is what precision medicine can deliver, and there's what it is that it cannot deliver. The example I gave in the neighborhoods of Chicago, for example, precision medicine cannot do anything about that. And we need to appreciate that. Otherwise, we will set ourselves up for failure, and the public will say, oh, you said precision medicine is coming to cure everything, but it is not going to do so. So thank you very much, and look forward to questions later on.
Great. Thank you, Dr. Rotimi. We're going to hold questions and comments for Dr. Rotimi until Dr. Burke has given her talk. Please go ahead, Dr. Burke.
Thank you very much. I really appreciate the chance to be here. And I guess I need a little help getting my slides up.
Dr. Burke, if you just click on the presentation and then press the page down, and your slides will be right after Dr. Rotimi.
Okay. Great. Okay. Thanks very much. So I am going to build on some of the very important points that Dr. Rotimi has made, but I want to start with just a comment that there is a discourse around precision medicine, genomics, and health disparities that I think you can already see from Dr. Rotimi's remarks over promises. So we see claims that precision medicine will give everyone the best chance of good health. It will improve outcomes for communities with disparities in health and help us to understand the complex interplay that creates health disparities. And I want to argue that the reality is more complex, more nuanced, and leads us, I think, to be cautious and careful about what genomics can offer us and what the role of genomics is in the issue of health disparities. And a fundamental point already very well made by Dr. Rotimi is the importance of social conditions as fundamental causes of disease. The fact that disadvantage creates conditions that put people at risk of risks, as Lincoln Felon have so well said. In other words, in disadvantage, people living in disadvantage experience multiple risk factors. They experience disparities in multiple different disease conditions, culminating, as Dr. Rotimi has shown us, in striking differences in life expectancy. And the important point here is that disadvantage maintains an association with disease even when the diseases that dominate disparities change, so that people living in social disadvantage in the early 1900s had higher rates of infectious disease. Today they have higher rates of diabetes. The unifying factor here is social disadvantage. So I'm going to go through 3 examples to get at what I think are the complexities of this issue and help us to think about where genomics has a role in this area of research. And I want to start with acute lymphoblastic leukemia. So this is a classic example of a health outcome disparity. What you see in this slide is data showing that the survival rate for African American children, Native American children, and Latino children is distinctly lower than that of white and Asian children. This is a disparity that has been documented for some time and persists. And it has certainly been suggested that genetics may play a role. What's really interesting when you start looking at this question is that the actual incidence of childhood acute lymphoblastic leukemia is much lower in African American children than it is in white children. And I'm going to focus on that black-white difference for a moment. So that kind of goes against our notion of health disparity. That is, we tend to think about health disparities as disparities in the burden of disease. But what we're actually seeing in African American children is that they have a lower incidence of disease, but when they get the disease, they have a higher mortality. And why would that be? First of all, genetic contributors are not an explanation. Sorry, let me start over again. African American children have a lower incidence of acute lymphoblastic leukemia, and genetics appears to be the reason for that. So the lower incidence, going contrary to what we might expect for health disparity, is probably due to genetic differences that lead to fewer cases of acute lymphoblastic leukemia in African American children. On the other hand, we do see that African American children tend to have a higher rate of poor prognostic indicators for several measures that are listed here. These are measures in children diagnosed with acute lymphoblastic leukemia that are associated with poorer outcomes. And so one possible interpretation of this would be lower incidence, higher mortality rate because there's simply a different kind of acute lymphoblastic leukemia going on, and maybe that's due to genetic differences. In fact, we don't really have evidence to say that genetics is a cause of those poor prognostic indicators, and we do have some hints that environmental exposures may be a factor, and those include environmental exposures in the parents' workplace, so parents' exposure to pesticides, and possibly also residential exposures, in particular related to processing of gas and other fuels. Poor outcomes, poor prognostic indicators may be a factor, even though there's a lower incidence. But the most important finding that we have that I think helps us to begin to understand what's going on here is the results of therapy for acute lymphoblastic leukemia in Black and white children at St. Jude's Hospital. What you see here is both in terms of 5-year and 10-year survival, there is no difference. Even though these Black children had the same poorer prognostic indicators on average than the white children. There's something very interesting about the care that's provided at St. Jude's. It involves aggressive case management, making sure, for example, that kids get all their follow-up care after they leave the hospital, and it involves no out-of-pocket costs for parents. Insurance is billed, but any additional costs are covered. And this very striking observation tells us, I think, that the poorer mortality outcomes observed in African American children as compared to white children with acute lymphoblastic leukemia are very much a factor of social conditions, social environment, opportunities for accessing good quality care. This is also, as you can see, not a health disparity in a traditional term. That is, it's not a difference in prevalence of a disease. It's a health care disparity that can be solved by good quality care. Now, let me talk about another example, type 2 diabetes. Type 2 diabetes accounts for most of diabetes, and genetics plays an important role. It's estimated that variants, more than 100 gene variants contribute to risk, all with small additive effects. And importantly, in studies of ancestral groups, we see differences in variant prevalence. We see some variants that are found exclusively or predominantly in specific racial or ethnic groups. And that has led to the speculation that genetics accounts for disparities in prevalence of diabetes. In particular, could genetics account for the very high rate of diabetes that's seen in the Arizona Pima? The Arizona Pima, in contrast to population rates on the order of 10%, have a prevalence of type 2 diabetes of about 37–38%, a very striking high prevalence of diabetes. And genetics has been a major contender for causing this disparity. A recent study of two Pima populations, one, the Arizona Pima with the high rate of diabetes, and another Pima population in the Sierra Madre Mountains of Mexico, has shed some very important light on this disparity. These two Pima populations share common ancestry, distinct from other Native American groups, with documentation of very high genetic relatedness by genomic markers. And yet, they have a strikingly different prevalence of diabetes. So you see in the U.S. Pima that very high 37 to 38% average prevalence of type 2 diabetes, where in the Mexican Pima, the prevalence is 6 to 8%. So we can't look at this and say genetics is the cause for that high U.S. Pima diabetes rate, because genetically the Mexican Pima and the U.S. Pima are very similar. However, the two populations are very different in another way. The physical activity rate is much higher amongst both men and women in the Mexican Pima compared to the Arizona Pima, and rates of obesity are much higher in both men and women among the U.S. Pima as compared to the Mexican Pima. And of course, these are factors that are well known as risk factors for type 2 diabetes. So I think we're led to the logical conclusion that the strikingly high rate of diabetes in the Arizona Pima is due to these social environment factors, rates of physical activity and rates of obesity. And it's worth then asking, how did the Pima lifestyle in Arizona change with Western settlement? So as white settlement came into the region where the Pima were living, native agricultural economy was disrupted. There was diversion of water to white settlements, and that resulted in simply the loss of agricultural land, of effective agricultural land. This led to the loss of healthy traditional foods, to poverty, to the use of surplus commodities as dietary mainstays, and concurrent with these changes, we see communities experiencing cultural loss, stigma, discrimination. So here we have an interesting example of a clear health disparity, one that has been speculated to be genetic in cause, but in fact can be explained by social factors, by social disadvantage, basically. Now, let's go back to this, this diagram looking at the differences in diabetes rate between Mexican Pima and U.S. Pima. What's interesting about this diagram is that they also compared diabetes rates between the Mexican Pima and their non-Pima neighbors. And you can see it's not statistically significant. The confidence intervals are overlapping, but there's at least a suggestion that the rate of diabetes is lower in the non-Pima Mexicans compared to the Mexican Pima. There's not enough power to really establish that difference. My point here is that I think we can account for the difference between the Mexican Pima and U.S. Pima rates of diabetes by social factors. We cannot rule out the possibility that there may be some more genetic predisposition amongst the Pima toward diabetes than perhaps other populations. In other words, we know that genes contribute to diabetes risk. We know that genetic variation may vary amongst populations. It is certainly possible that there are differences, probably modest differences from one population to another in the likelihood that diabetes will develop. But it's the social factors that create the striking difference, the true health disparity. And now I'm going to talk about the example that Dr. Rotimi has already introduced, and I want to talk about what I think are some of the complexities here for the topic that we're discussing today. So African Americans experience a disproportionate burden of chronic kidney disease and end-stage kidney disease, and as Dr. Rotimi has already shown us, risk variants in the APOL1 gene contribute to increased risk. There's a risk genotype associated with increased risk that is present in 13% of African Americans, and the penetrance is estimated at 20%. And I want to linger there for just a minute. Penetrance of 20% means that there's a very substantial risk if you have this risk genotype that you will develop kidney disease. But a penetrance of 20% also means that there are important modifiers. It's not 100%, it's 20%. So there are other factors that influence whether or not someone with the risk genotype is going to develop kidney disease. And as we think about the implications of this particular genetic risk factor, clearly it's going to be very important to think about what might be the modifiers. We have a very interesting study from the cardiac cohort, which has followed white and black populations over a long period of time. And this particular study had the goal of shedding light on the risk associated with APOL1 risk. So we're looking at data from the cardiac cohort following individuals who did not have kidney disease at any evidence of proteinuria, albuminuria, which is an indicator of chronic kidney disease at the start of the study, and then following them over time. And the study group has been divided into white and then low-risk black and high-risk black. Low-risk black means individuals don't have the APOL risk genotype. High-risk black means they have the APOL risk genotype. And as you can see, the risk of developing chronic kidney disease is progressively higher when we compare the white population to the low-risk black population, which is more than twofold higher, to the high-risk black population, which is more than fivefold higher in its risk of developing chronic kidney disease compared to the white population. What's really interesting here is when we correct for smoking, blood pressure, high mass index, diabetes, socioeconomic status, the difference between white and
Dr. Burke, you're cutting out slightly.
Oh, sorry. Can you hear me now?
You're very faint.
Huh. Okay. How is that?
That's perfect.
Better? Sorry.
Yes.
I'm trying to, put my voice right up to the phone. Okay. So what we see is when we correct for a variety of risk factors that are associated with socioeconomic disadvantage, the difference between risk in white and low-risk black seems to go away. We certainly still have a significant risk from the APOL genotype. So the APOL1 genotype is a very important contributor to risk, and it is presumably a genetic contributor to a health disparity. Very unusual, but important. It is not the only contributor to this disparity because we see that there's a substantial difference between low-risk black individuals and white individuals in their rate. But now I want to talk about what we know about APOL1 and its effect on kidney disease. And that is that the effect on kidney disease seems to be primarily an effect on the rate of decline in kidney function after the original insult. So what we're seeing in this diagram is the gray boxes indicate the mean age at which albuminuria began. That is the evidence of chronic kidney disease, and there are differences, but the really dramatic difference is the rate of decline that occurs in the people with the APOL1 genotype, the high-risk Black people, after they have developed evidence of kidney insult. And what that raises as a very serious question is whether efforts to protect individuals from the effects of kidney risk factors, things like early hypertension, things like developmental exposures that may affect kidney development, whether those kinds of measures might perhaps not only be helpful to low-risk Black population or to other socially disadvantaged populations, but also to high-risk Black populations. In other words, there is a possibility that the penetrance of 20% that we see for APOL1 could be reduced even further in individuals with the APOL1 risk if they were subjected, if they were exposed to a more advantageous social environment, one that was more protective against other risk factors for renal disease. Now, I can't conclude these remarks without saying it obviously is extremely important to continue doing the research to understand the mechanisms behind this APOL1 effect. I can only underscore what Dr. Rotimi has said about the importance of genomic research in that kind of context. But I think this example says to us that it's equally important to understand the social determinants that contribute to kidney risk. And I will just end by saying that it is crucially important that we place genomics in context. Genomics may explain some population differences in disease instance. Sometimes those differences will be aligned with the outcome disparity, as in the case of APOL1, but sometimes they won't be, as in the case of ALL. More importantly, if we study genomics in isolation, we run the risk of obscuring far more important social determinants of health, and in so doing, we have the risk that we will stigmatize disadvantaged populations, actually propose to them that it's genetics rather than social disadvantage that's causing their problems, and ultimately slow progress toward the very important goal of diminishing health disparities. Thanks very much.
Okay. Thank you, Riley and Charles. This is Maureen Corey, Director of the CDC Office of Public Health Genomics. We have about 11 minutes for discussion. I would like to invite everyone. We have more than 300 people registered, or about 300 on the web. If you want to submit questions, we may have already a couple of questions. People in the room, you want to line up your questions, write them down, send them to me. I'd like to start off with a sort of a more generic discussion, and you both, at one point I thought you were disagreeing with each other, but I think you're very much in agreement. This webinar series is about building a bridge between precision medicine and population health. One major concern for population health is health disparities, and it's very clear from both your presentations that health disparities is accounted for by as much environmental or social determinants of health and genetic factors. So one of the driving forces of precision medicine has been genomics, but if you look at the NIH Precision Medicine Initiative and where we are with the current thinking, it's more putting the genetics in the context, as Riley said, looking at the environment, looking at the lifestyles, looking at social determinants. So the question I have for both of you is, so what can precision medicine broadly defined, which is looking at genes and environment and lifestyle, do on the short run for population health, which is a big question mark, but along the way, where are the low-hanging fruits in terms of reducing the health disparities? So it's not a question of either genes or environment, but looking holistically at genes and environment. So who wants to go first? Charles, you're in the room.
I will give it a shot. I think the example of sickle cell with gene editing technology, I think is a very, very good one in the sense that that is clearly a genetic disease by any definition of the phrase, and is lending itself to the new genomic technology of gene editing. And we are just at the beginning stage. There's been a successful experiment done in mouse that shows that we can indeed edit this gene and cure this condition, which is devastating in African and African ancestry populations globally. So that is clearly a good example. In terms of precision medicine, though, I think what we are trying to, my take on precision medicine is, I look at it as a sample size of one. And that is, you are sitting in front of your doctor, you and your doctor, and somebody is trying to prescribe a medicine for you. Right now, if it's hypertension, most likely if you're African ancestry individual, they give you a diuretic to start with. And if that doesn't just work enough, they might put you on an ACE inhibitor or something else and keep going down the road. Wouldn't it be nice at some point that we don't need to go through that stages and be able to say, oh, if Charles Rotimi walks into a clinic, we can interrogate Charles Rotimi genome. It's already part of his driver's license. And we can put this into something to read it and say, oh, we know that Charles will respond very well to ACE inhibitor as opposed to starting him on a diuretic or testing 3 drugs. To me, that is where we're going. And in that instance, some of this issue about skin color, who is black, who is white, really does go away, falls apart, because I may carry that variant, my sister may not carry it. Even my mother may not carry it. So that, to me, is where precision medicine should be taking us. If we allow it, if we stop using this kind of racial labels or skin color, in terms of how we design the study and how we interpret our data.
Thank you, Charles. Wiley, you get the shot at this.
So I will start by saying that I think if we ask the question, what should we be doing for health disparities, it's very clear. Children should not go hungry. People should live in safe environments. We should increase the quality and access of health care. I don't think the top approach to health disparities is mysterious. I agree that there may be some very interesting precision health research that could refine some health disparities. For example, why and how is it that shift work is associated with bad health outcomes, that sort of thing. It may be that molecular techniques can help us to understand the mechanisms by which social disadvantage is embodied. But I think our approach to health disparities is really the, well, let me say that the measures that would address health disparities are pretty straightforward, and they don't involve genetics. So the question really, when genetics comes in, it comes in 2 ways. One is, as Dr. Rotimi has outlined, in the healthcare of the future, we want to make sure that everyone can benefit. Now, let's start by saying, in the healthcare of the future, we'd like everybody to have access to healthcare, to good quality healthcare, to available and affordable healthcare. But we hope that healthcare will include some benefits that will be provided by genomics, like pharmacogenetic tests that will improve the quality of the safety and the efficacy of certain drug treatments. And the only way we'll get there is by being inclusive in our research. I think genomics has a role, a small role, I would say, in addressing future health care disparities. And it clearly has a role, and I think that's where the sickle cell disease example comes. It has a role in addressing rare genetic diseases suffered by populations that live in social disadvantage. We'd like to see as much progress being made on sickle cell disease as on cystic fibrosis, for example. But the big picture is that health disparities is really not about genetics.
Okay. Thank you, Riley. Thank you, Charles. I'm looking at some of the questions from the web and here in the room. I have at least 4 or 5 questions on epigenetics. Epigenetics is a hot topic these days, and it kind of embodies the idea of gene-environment interaction and how social influences or environmental influences can play a role on the gene, in terms of gene changes over generations. So do you have any, I didn't hear you mention epigenetics in either one of the talks. Do you have any thoughts about the role of epigenetics in the use of, in either understanding health disparities and, closing the gap in health disparities in the futures and understanding biological mechanisms and so on and so forth? Wiley, you can go first because I know you have worked on this. Yeah, go ahead.
Yes. Well, I think epigenetic research is very promising for understanding that fundamental issue of how social disadvantage is embodied. So it's very likely, for example, that epigenetic changes in our circadian rhythm have some health impacts for people who do shift work, to cite that previous example. There's a very promising body of research around epigenetic changes in utero and in early childhood related to social disadvantage that may have long-term effects, as you say, not just for the individual, but possibly for that individual's children. So I think that's an extremely important area of research, and I think it will help us to understand better what's happening as a result of social disadvantage. Whether or not it will point us to different policies is, I think, quite unclear. In other words, our fundamental social policy should be to minimize social disadvantage, and particularly to raise kids in environments that are safe with adequate food, adequate access to education and health care, et cetera. And although epigenetics may help us to understand what happens when those conditions aren't present, I'm not sure it's going to point us to different policies.
Yeah. So, Charles, you don't have to answer this because I have 6 other questions. Okay. Just say you're in agreement with Wiley. Or you have other thoughts on this?
No, I think for the most part in agreement, but I just wanted to say epigenetics, I think the promise is going to give us insight into mechanism. Yes.
Okay. So I have a bunch of questions here, and I think things are converging along different lines, but this is one intriguing one. So this came from the room. In order to effectively utilize genetics to determine disease treatment, I would add prevention to it. Is it first important to look at racial or ancestry groups to identify risk genotypes? Then later, once the databases have that information, then personalized medicine can be applied? Question mark. And if the person who wrote that question has anything to add to it, Charles, you go.
Again, like the example I pointed out in terms of heart disease, it is critically important that as we move down this road of using genomics in healthcare, in understanding disease etiology, that we adequately represent the genetic variation that exists globally. It is only by doing that, by creating that comprehensive reference database, that we will be able to know what is disease, what is not disease, and also what is the prevalence of a particular type of variant, whether it's present or absent in certain groups. It can become actually quite instructive at a certain point, because you can imagine a nation that doesn't have a lot of resources in terms of their healthcare budget. They could, for example, say this particular drug is under the influence of this metabolizing enzyme, And we don't have this frequency in our population. It's at very low. Therefore, we are not going to buy this drug. We won't spend our minimal resources on this drug and do try and error. So you can see how that can inform even a national decision to which drug to buy or not to buy. So again, I think we have to adequately study our human population and capture the genetic variation that exists if we are going to do this correctly.
Okay. Wiley, do you want to add anything, or shall I move on to the next question?
Sure, go ahead and move on.
Okay, so it's 4 o'clock, and I'm going to allow a couple of minutes over time. People who want to leave, that's fine. I think the web will not just cut off on us, right? So I think this is, from the web questions, a lot on epigenetics, a lot on social determinants of health and the environment. I think that those have been explored. But the one that is intriguing to me, and I think, Charles, you may have answered this already, but I want to give it another shot, that some have suggested that genomic research in low, middle resource areas or countries should really be placed at lower priority given these other issues around health and disparities due to infrastructure, resource concerns, and your thoughts about how to elevate that genomic research in these populations. And you've kind of done it in the African arena, but what would be the answer if you would say, given that genetics is not important as a driver of population health, why should we invest in genetics research?
Again, I think when we make assessment about a population, what a population can afford or not afford or what is important, we need to be careful because that assessment could be very, very wrong. And even among people who think they really know. And the example that I usually use in this is cell phones. When cell phones first came out, people say, oh, don't bother to take it to Africa. They can't afford it. They won't be able to use it. But today we know that Africans use cell phone more than in the United States. And the banking infrastructure are now based on cell phones. So what makes us think that we cannot come up with a technology, some of it is actually under development, that is genomic-based, that can be used for newborn screening? That is currently not implementable in a lot of African countries. And if this technology is designed properly, it may not need electricity, it may not need refrigeration, and it could be pennies instead of the tremendous cost that it is right now that we're using to screen newborns. Yeah. So that would be my take on that, is that if most of the world, anyway you bet against technology, you tend to lose. So we are just beginning to appreciate how genomics is going to drive some of these health technologies, and we need to give it room, and we need to make sure we engage all global populations to make sure that we understand their own limitations and constraints.
Thank you. Wiley, anything you want to add?
I will just say that if a policy-making body came to me and said, our choice is to provide good quality primary care to everyone in our population versus pursue cutting-edge genomic research, I'd say, sure, you need to provide that quality health care. The issue is that, of course, there are fundamentals that must be provided and that are important and that often are not provided, which is really the underlying cause of health disparities. So I don't think we can ever say that those fundamentals aren't of the highest priority. Hopefully, in many places, and certainly in the U.S., we have the luxury that we will also have resources to pursue the cutting-edge research. I think that means that we should, and I think pursuant to some of the remarks Dr. Rotimi has made, make sure that our cutting-edge research is very inclusive.
Okay. Thank you. Thank you both. I would like to bring this session to a close and thank both our speakers for a tremendous group of presentations and good questions. Everything will be up on the web soon. And thanks again, and we'll look forward to another one soon. Thank you.